So this lesson is going to be focused primarily on the structure of DNA. And in the following lessons, I'm going to be talking about protein synthesis and many other aspects related to DNA. But for now, this is going to be a simple lesson on the structure of DNA. So DNA is a polymer which is composed of many monomers which are known as nucleotides. So as you can see, this is a DNA molecule. It's composed of two strands. You have the first strand here, and you have an anti-parallel strand here. And the overall shape of the DNA molecule is a double helix. So DNA stands for deoxyribonucleic acid, and that's mainly because of the composition of a nucleotide. And one of the three parts of a nucleotide is a deoxyribose sugar. That's probably why DNA is known as deoxyribonucleic acid. One of the reasons for that is because it is composed of a 5-carbon sugar. So 4 of the carbons form the sort of ring shape of the deoxyribose sugar, and the 5th carbon is part of the compound HOCH2. And I'll find a picture of that. Deoxyribose sugar. So this is what deoxyribose sugar looks like, and ribose would be present in RNA, while deoxyribose is present in DNA. So we're mainly going to be focusing on DNA. So a nucleotide is really composed of 3 things. The first thing is a phosphate group. The second thing is a deoxyribose sugar, as I mentioned, and the third thing is a nitrogenous base. So there are 4 nitrogenous bases that are classified under 2 subcategories: the pyrimidines, which contain a single ring, or purines, which contain a double ring. So the pyrimidines are cytosine and thymine, C and T, and the purines are adenine and guanine. So there are a few equations that are important to DNA. Really, there is one general equation for a nucleoside, which is a nitrogenous base plus a sugar. So let's find that. So a nitrogenous base would be, for example, guanine plus this 5-carbon sugar. This forms a nucleoside plus water, or H2O. And this nucleoside formula really applies to any of these N-base plus sugar combinations. So an equation of a nucleotide. So far we know a nucleoside is composed of N-base plus sugar. So a nucleoside plus a phosphate would really have all 3 components of a nucleotide. It would be the nitrogenous base, the sugar, and the phosphate. And so the general form of a nucleotide would be a nucleoside composed of the N-base and the sugar, plus a phosphate, which creates a nucleotide and water. So really it would just be the N-base here, plus the sugar, plus the phosphate. This 3 would create a nucleotide. I actually neglected to mention an example of a nucleoside. So we could have adenine plus sugar would create adenosine plus water, and for a nucleotide we would have adenosine plus phosphate would give us adenosine monophosphate plus water. So there's a really interesting way to sort of name the carbons that surround the deoxyribose sugar. So over here, just give it a second to load, we have 4 carbons that are really part of this ring, and then we have a 5th carbon in this compound. So we call this 1', 2', 3', 4', and 5'. and we have an OH here. The terminal OH is on the 3'. We have another OH on the 1', but the terminal OH is on the 3', and we have an H on the 2'. So really, the name deoxyribose comes from the fact that this H here is missing an O to create a hydroxide. So now let's look back at really the structure of the DNA. This is sort of a pictorial representation of the structure of DNA. Let's look at some of the bonds that are formed. So you can see double bonds between the double hydrogen bonds between the adenine and the thymine, and triple hydrogen bonds between the guanine and the cytosine. So from this we can tell that the guanine-cytosine bonds are stronger than the adenine-thymine bonds, and these hydrogen bonds are also known as dipole-dipole bonds. And really how it works is it's based on the Linus-Pauling electronegativity scale. So as you go to the right of the periodic table and as you go up the periodic table, electronegativity increases. And since hydrogen has a lower electronegativity than oxygen, hydrogen has a smaller pull on the electrons when bonded to oxygen, which means that it has a slightly positive charge compared to the slightly negative charge on oxygen. Let me find an image of this. So, as you can see here, let's probably choose this one. This sort of symbol represents a partial charge, so it's not ionic in that electrons are being transferred, nor is it covalent in which they're shared. It's somewhere in between that. Oxygen atom is partially negative, while the hydrogen atoms are partially positively charged. So yeah, that's what a dipole-dipole bond or dipole-dipole force is. Okay, so there's another kind of bond. This, you see, these phosphate groups are attached to the nitrogenous bases, and this is really what is called the backbone of the DNA molecule. This phosphate sugar linkage, as we can see, this one is bonded to the 5'. This phosphate group is bonded to the 5' of this sugar, while and it does 5', 3', 5', 3', so on until 3'. And the reason why this backbone is anti-parallel to this backbone is because this phosphate sugar sort of combination starts from 5', goes to 3', while this backbone starts from 3' and goes to 5'. That's why it's called anti-parallel. That's sort of where that word is derived from. So as you can see, these anti-parallel sort of backbones have bonds between the sugar and the phosphate, and this is a covalent bond that's known as a phosphodiester linkage in the sp-sp-sp. So there's also another kind of bond in which the nitrogenous base is attached to the 1' carbon of the sugar. So a case for this would be you have the 1' carbon of the sugar would be this one, and the nitrogenous base would be this. So there's really a glycosyl bond between the 1' carbon of the sugar and the nitrogenous base. So far I've listed 3 bonds that are happening. There's a phosphodiester linkage between the phosphate and the sugar, which is a covalent bond. You have a glycosyl bond between the 1' carbon of the sugar and the nitrogenous base, and a hydrogen bond, which is a dipole-dipole bond between the nitrogenous bases. And we have double hydrogen bonds and triple hydrogen bonds, depending on the base pairing of the nitrogenous bases. So what can we talk about now? We can talk about, okay, perfect. Let's talk about base pairing. So base pairing is the pairing of a purine, which I mentioned is a double ring, with a pyrimidine, which is a single ring. And this is what is known as complementary base pairing. So there's very easy rules for this. When you have a thymine, it'll always be bonded to a adenine, and when you have a guanine, it will always be bound to a cytosine, and vice versa. So if you have one strand, not one strand, but one side of nitrogenous bases, let's say you have thymine, adenine, cytosine, and guanine, you can easily anticipate the other side, just because of the complementary base pairing rule. So you have a thymine, you know there's going to be adenine here. You have an adenine, you know there's going to be thymine, and vice versa. You can continue down here. So really, I just think of it as ATGC. So that's the pairing rule, and this is what is known as Chargaff's rule. The reason it works is that the amount of adenine, for example, is always, is very similar, or if not equal, to the amount of thymine, and the amount of guanine is very similar to the amount of cytosine, if not equal. And this is what's known as Chargaff's rule, as I mentioned. So that's nearly the end of the DNA structure lesson that we had in class. I think hopefully I clarified some of the confusion regarding some of the terms, some of the nitty-gritty details. But to finish the lesson off, DNA was first determined in 1953 by two scientists, James Watson and Francis Crick. Although there is a lot of speculation regarding whether or not they were really the first ones to discover the double helix structure. There's another scientist by the name of Rosalind Franklin, who supposedly used X-ray diffraction to negate the idea of a triple helix structure, and that gave way to the double helix structure. And Watson and Crick eventually ended up getting a Nobel Prize for that discovery, while Rosalind Franklin unfortunately passed away, and the Nobel Prize isn't given posthumously. And supposedly Watson and Crick didn't publish the discovery first, and didn't even give sufficient credit to Rosalind Franklin. But okay, well that's about it. Let's just based on what the textbook says, let's give the credit to Watson and Crick. So I mentioned the fact that these are anti-parallel, because this goes from 5' to 3', this goes from 3' to 5', and some facts regarding the double helix structure of DNA. The diameter is 2 nanometers, and a complete helical turn is 3.4 nanometers. And some other information is that the purine and pyrimidine bases, the nitrogenous bases, they are bonded to a deoxyribose sugar through a nucleoside thing, and they project inwards towards the axis of the helix along the same plane. It really reminds me of some of the rules of light. What was it? Forget it. Okay. Well, if you thought that that lesson was informative, you thought that it really cleared up some ambiguities that you had, or some things that you didn't understand about DNA structurally speaking, you can just subscribe to my channel, or like the video, or share it. Anything would be great. And in the next lesson, I'll be talking about DNA packaging and DNA replication and protein synthesis, and things along that line. So thank you for watching.